This is CBE 205 project for Saad Ahmed and placing. Our project will be about genetic engineering. First, we will talk about the history of the genetic engineering. The term genetic engineering was first used by Jack Williamson in 1951 in one of his famous novels. Then in 1980s, scientists discovered accurate ways to modify genes, which was a major breakthrough in biology. Uh, the first genetically engineered plant was tobacco, and in the next slide, I would talk more about tobacco. It is used as an example of it, especially in medicine. In 1965, uh, Mendel discovered the basic rule of inheritance, which became the foundation of genetics. And in 1953, Worston and Crick described the structure of DNA. Also in 1972, Powell creates the first recomponent of DNA. Finally, in 1973, Cohen and Pure transfer genes between organisms, marking the first genes cloning experiment. Here I have some famous breakthroughs. In 1981, for example, the Food and Drugs Administration approves the first genetically engineered drugs, which famous as insulin. And in 1994, the flavor saver tomato became the first genetically modified food. In 1996, Dolly the sheep is cloned Chown case somatic cell nuclear transfer. Finally, in 2003, was the completion of a human genome project, mapping all human gene. Here, we would talk about the genetic engineering in the modern era. In 2003, CRISPR introduced as an accurate gene editing tool. Gene therapy is a process is making a process in treating genetic disease like sickle cell anemia and cancer. Also, synthetic biology involves designing new organisms for medicine and sustainability. Finally, they care about the ethical concerns over biodiversity and human genome editing and united consequences. And that was in 2020. Second, I'm going to talk about the benefits of genetic engineering and its applications. First, I have the definition of genetic engineering, which is a process that uses laboratory piece technologies to make a change in the DNA of a specific organism. Here I have some uses in three different fields and some examples for each one. Agriculture, for examples, palm tree and rice and European corn as well. For medicine, they use tobacco, as I mentioned earlier, to induce the amino response. Or for industry, they use enzymes with desired properties and biofuels production as well. Here also I have general benefits of genetic engineering. For example, improve the crop yields and quality as well, introduce herbicide resistance that result in less herbicides being used, which end up with more yields killed, and that's going to be faster. Also, making trial insects like mosquito, which would lead in the end to reduce a disease like malaria. And finally, genetic engineering would assist us to have faster result than selective breeding in some organisms. Finally, I would talk about how can genetic engineering be used as a treatment. Gene therapy, which is a variation of genetic engineering that involves replacing editing, what would be responsible for disease development, to defective the genes, and that's going to be responsible for disease development. Also, gene therapy can be used to modify uh, person genes or cure some disease and illness like HIV, AIDS, sickle cell anemia, and sites fibers, etc. But I'm gonna go deep with the HIV, how gene therapy works to treat HIV. I have a picture here that's gonna make it more easier. We have some steps. The most common technique is ex vivo, which is outside the living organisms gene therapy, and that's going to be done by a few steps. First, collecting the patient's cell through bone or blood marrow to isolate the T cells. Second, modify the cells in the laboratory by adding a gene editing tool into the isolated cell. Third, expanding the modified cells in the large numbers to ensure there is enough for an infusion. And finally, in infusion, the modified, which is HIV resistant here, cells back into the patient's bloodstream where they multiply and replace any weak and infected cell. In the end, I hope that we give important points about genetic engineering, and I hope our slide was clear. And I would love to say thank you for listening.